So backcross breeding is when you have a wild species that is going to be a donor plant. And that donor plant is going to be ugly, problematic. It's the wild germplasm, the wild weed, but it's got something I really, really want. One or two genes. That's all it can have that I want. And so what do? I take my wild donor, A prime, that has one thing I really want, and then I cross it with my elite variety, the variety that has everything I want except that one disease resistance or that one whatever it is. And I cross them and I get my first filial generation, half elite variety, half wild donor. That's too much to be 50-50. This is going to be an ugly plant that's useless. So what do? I take the F1 and I cross it back to that mother plant, or at least a clone of that mother plant, the original elite variety. I cross it back to the same elite variety again. And I get my, instead of F2, it's now called backcross 1 because it distinguishes that I crossed it again with that elite mother plant. So I cross it again, backcross 1. I cross it again, backcross 2. And depending on what you're trying to do, you might go to backcross 3, 4, 5, 6, 7, 8. Takes time, but I want to get rid of most of the genes from this wild species and only have the gene I'm looking for. If I have good knowledge about that gene, I can test it at every single stage. Not the F1, that's not important. But at the first backcross, let's say I'm looking for disease resistance. What am I going to backcross? I grow up my seeds. I spray them all with mold. Anything that dies doesn't go to backcross 2. Anything that lives goes to backcross 2. And then I grow up my backcross 2 seed. Spray them with my mold spores. Anything that dies, bye, didn't have the disease resistance. Anything that lives, backcross 3. And I keep doing that until I'm satisfied that I have the disease resistance that I was looking for, or whatever it was I was looking for. You need some way to test for it. So I have that, and I've gotten rid of those extra baggage genes that made the weed look like a weed. So at this stage, what is it? 50% weed, 25% on average, 25% weed. On average, about 12.5% weed. That's still a lot of weed. So beef backcross 3, backcross 4, 6%, 3%. Now at 3%, maybe you're doing fine. Maybe that's good enough. Maybe I got to bring it down to about 1%. It depends. But every time I cross back, I cut that in half, the amount of weedy genes. And so that's great for taking a single gene from an unadapted wild or weedy species, unadapted species, into an elite variety. My perfect variety needs one thing. Backcross breeding is the way to go. And this is actually one of the staple techniques of most or many breeding programs. And then there's a variation, and it's a variation that I'm not going to get into detail, but I do want you to at least have seen it. It's called the inbred backcross breeding. It's like combining backcross breeding and single seed descent. And so it starts off just like backcross. I take my recurrent parent, my elite parent, all right? So if you're writing this down, taking your own notes, the recurrent parent is my elite variety. My donor parent is my weed. And I get my F1. I cross it with my recurrent parent to get my backcross 1. That's still too much. That's 25% weedy species. And then I go to my backcross 2. I cross it back again. I go to backcross 2. And at back cross 2, I'm at about 12%. Back cross 3, I'm at about 6%. These are manageable amounts. And so I take maybe 60 to 150 individual plants from the back cross 2 and back cross 3 that now have a big chunk of your elite recurrent parent and a smaller chunk, 3 to 12% ish of the donor parent. Because remember, it's averages we're talking about. The individual plants are going to vary. This is 50, but this is averaging 25% weed at the back cross 1, averaging 12.5% at the back cross 2. You'll have some with a little more, some with a little less, because they're mixing it up. And now that I've got to the back cross 2 and 3, I pretend like I start fresh. I've got my back cross 2 seed. I take 60 of them, and I start doing single seed descent. And this is single seed descent going off to the right in the bottom portion. And I do a set of single seed descent breeding on the back cross 2 generation and another set on the back cross 3 generation. Depending on the math and what you're trying to do, you might even do a set on the back cross 4, 3 and 4, but 2 and 3 is probably the sweet spot for what you're trying to do here. And so this is when you're trying to do multiple genes from that wild species. Remember, back cross breeding gives you 1 gene, maybe 2. What if I need 4? That's really tough. And so this actually started out as more of a population genetics mathematical theory. Somebody was just studying the math of population genetics. It wasn't about plant breeding at all. And this was way back in the 1950s. And one of the professors I had yanked this out of the 1950s archives and started looking at it and saying, that might actually be good for breeding wild plants that we need multiple genes from it. And he was right. It now is a common technique that's used in certain crops. And I used it for my PhD thesis, actually. This type of breeding was exactly the type of breeding I used for my thesis in tomatoes. So plants and animals have a complex cell cycle. The cell cycle is going from cells growing to dividing. And then once they've divided, they're small again, and then they have to grow. When they get to a certain size, they divide again. And that's how we heal, that's how we grow. And there are 2 ways that cells can divide. Most cells divide by something called mitosis. Mitosis is all of the somatic cells, the non-reproductive cells. In us, it's everything from skin, hair, heart, liver, brain, you name it, our body, everything except the reproductive cells being created in ovaries or testicles. That's the only thing that does not divide by mitosis. And even the major cells of the ovaries and the testicles, they divide by mitosis too. It's only those cells that are dividing to make eggs or sperm that divide by the second type, meiosis. Now, unlike meiosis, mitosis keeps all of the same number of chromosomes. Every cell is a clone of the previous cell. Now, they might go off in different pathways of what they're doing, so they're genetically the same, but one cell becomes a liver cell, another cell becomes an eye cell or whatever. So they can take on different functions, but they genetically are clones. In meiosis, that's not true. These are cell divisions that occur in what's called the germ cells or reproductive cells. And they produce gametes. There's 2 types of gametes in all of the kingdoms of life. The gametes are eggs or sperm, one or the other. And in the process, meiosis halves the number of chromosomes, cuts them in half. Because if I have egg and sperm cells that are with the same number of chromosomes as the rest of the cells, and two of them come together, every generation your chromosomes are doubling. That's not okay. You can't double the chromosomes every single time. So what happens? What happens is simple. The chromosomes are cut in half. And I say that's simple. Yeah, it's easy. It's actually very, very complex. So here are the chromosomes in the human karyotype, the collection of all chromosomes. Now, this is a collage of many different pictures. Each of these individual chromosomes was photographed separately and put together in this karyotype collage. And humans have 23 pairs of homologous chromosomes. Homologous meaning they're the same chromosomes with the same list of genes, but they're not identical. They're not identical because they have different versions of those genes or different alleles. So I got one chromosome number 1 from mom, one chromosome 1 from dad. It's okay, babe. Okay, mommy will be back soon. Puppy's really upset that mommy left. Anyway, and I'm not paying attention to her either. She'd be fine if I was playing with her. So mom gave me one chromosome 1, dad gave me another chromosome 1, but they're different. They're homologous. They line up, they match. They're the same genetic information in general, but they have different versions of some of the genes. And those versions might be, well, mom maybe had green eye color and dad had brown eye color, things like that. They both had the eye color genes, just one had the green version and one had the brown version of that gene. So we call them homologous. We have 23 homologous pairs. So look at this chromosome karyotype of a human. Can you tell me what the person is? Can you tell me anything about the person in this karyotype? Anything? Can you tell that it is most likely a male? If you notice in the lower right-hand corner, there is an X and a Y chromosome. X and Y are the sex chromosomes, so they determine the gender of the person in most cases, but there are lots of exceptions. So the fact that this person has a Y chromosome means they're male. Now, everybody has to have at least one X. If you don't have one X chromosome, you can't survive. There's essential things. So there's no such thing as a YY human. That would never develop into a living person. You need the X. But females have two X's, males have one X and one Y in most cases. But there are some X's that have a gene on it that's called the TDF gene, or testosterone determining factor, and that's what makes you male. And so there can be XX males. And are they exactly a perfect typical male, or are they slightly different? They might be slightly different, although you might never know it. There are a lot of XX males that they go through their whole life and never know that they are an XX male. And there can even be an XY female. An XY female would have a Y chromosome that does not have a functional TDF gene on it. So they don't get the signal to turn male. Everybody's born female. Everybody is born female, or not born, is in utero during development, starts as a female. And at some point during gestation, the TDF gene kicks in and the plumbing's rerouted, and the fetus turns into a male during development if the TDF gene kicks in. Some people have asked me about hermaphrodites, and that is something that doesn't exist in humans, but there are some animals that are, many animals that are true male and female at the same time. That is a hermaphrodite. They can be both male and female. Snails are a great example that that's their normal functioning biology. They are hermaphroditic. And with humans, we don't have fully functioning male and female, but we have intersex, where they are humans that have characteristics of both male and female, but either only one of the sexual organ, reproductive organs are functional, or no reproductive organs are functional. It depends. And there's all kinds of variation with all kinds of different different expressions of that in what it means to be human. And one of the things that really bothers me is there's a lot of people who think that, you know, that something is obvious, as there are men and there are women, and that, you know, that is something simple, and whatever your belief system is, it is not that simple. And I don't care what your belief system is, except I don't understand how somebody could believe that somebody who is a perfectly functioning normal human being and possibly sitting right next to you and they are an intersex person, why don't they count in your philosophy? They should count. And we freak out about it in this country more than any other country that I've ever visited anyway. You know, there's this idea that everything's black and white, male and female, and it's like the real world. Mother Nature doesn't do that. Mother Nature's got all kinds of grays, even in areas where you think it wouldn't have gray. Get over it. And I hope you have room in your brain and your heart and your open mind that you realize that there are lots of people that are not strictly male, not strictly female. They're somewhere in between. And even if they have all of the genetics of male or female, there are other factors that go into play with sexual identity and other things that are very complicated. So enough on that soapbox. So how does mitosis actually work? There are stages of mitosis, and I'll keep it simple for this class. I go into it a lot more deeply in the plant science class, and I have higher expectations on knowing this stuff in my plant science class compared to the veg crops class. And no, I don't want whatever. My phone listens to what I say. It just thinks I asked it a question. Go away. Anyway, you'll notice this dotted line here. The dotted line separates the non-mitosis portions of the cell cycle from the mitosis. Everything on the right is mitosis. Everything on the left is not mitosis. So first we start with number one. The cell grows. It's got to reach its time for division. So we call this interphase, in between division, or intermission, you could think of it. It's in between cell divisions. And during that, there are actually three phases that are occurring in this interphase. A growth one, where the cell's getting bigger, the S phase, and the G2 phase, where there's DNA synthesis and preparation for cell division. And now prophase, the chromosomes condense, and you can see the chromosomes that have replicated already. So you still only have, in humans, you still only have the 23 pairs of chromosomes, but they are condensed and easier to see under high magnification. And they look like little X's because they're reproduced, they're copied. It's still considered one chromosome, even though there's twice the amount of DNA. It's not considered a new chromosome until they split. So prophase, chromosomes condense. Metaphase, the chromosomes line up. And so here I have 4 chromosomes in this picture. Number 3, 4 chromosomes, meta, middle. Metaphase, they line up on the middle, or the equatorial plane as it's called. Or central plane in this, but equatorial plane in other texts. And then they start to pull apart, anaphase, and there are these special protein handles on the centromeres, these little blobs in the center that hold the chromosome pieces together. And they're little handles and they get pulled apart. And as soon as they pull apart, now it's 2 chromosomes for each one of those. And it's like pulling, I liken it to a pool noodle, those long foam noodles. If you've ever been swimming in a pool, you drag the pool noodle through the water. What happens? It kind of folds back. It doesn't fold all the way back because it's a little bit rigid, but it folds. If my hand is here, it folds around my hand. And as I'm pulling it slowly through the water, it's kind of trailing back due to friction in the water. And the same thing is happening here as these chromosomes, these condensed chromosomes, are being pulled by special protein fibers that are pulling them to the sides. And you can see these little V formations. And so it's easy to spot under a microscope when you have a good picture of anaphase. You can see these V's being pulled in opposite directions. Telophase, the chromosomes collect at the opposite poles and a new nuclear envelope starts to form. And then cytokinesis, the cells split in 2. And you start all over. Now I got 2 small cells with exactly the same chromosome complement, clones of what I started with. And now they grow and do it again. That, my friends, is mitosis. Meiosis, on the other hand, is kind of like mitosis twice. So how does meiosis work in the germ cells? You start with a germ cell line, a cell that is about to divide to make an egg or sperm. So it is diploid, meaning it has 2 copies of the chromosome, mom and dad. And it's still the same as from mom and dad at this point. Prophase, the chromosomes condense. Metaphase, the chromosomes line up along the equatorial plane. Anaphase, the chromosomes are pulled apart and collect at opposite poles, but there's no nuclear envelope that forms in telophase. They just collect on the other side, and then it immediately goes into prophase 2. They stay condensed. Prophase 2 is not much happens. There are some things that are important, but for this class, not much happens. And then we go to metaphase again, anaphase again, and telophase. So it actually divides twice. 1 cell becomes 4. But in the first anaphase, the chromosomes line up in metaphase. In the first anaphase, I didn't split the replicated chromosomes at the centromere. I just separated the chromosome from mom and the chromosome for dad, sort of. Get into that in a minute. But I separate the chromosome pairs. And then I split the replicated chromosomes in the second anaphase. So I actually cut the chromosome number in half because I replicate once and I divide twice, effectively cutting the amount of chromosomes in half. And I end up with 4 haploid, half the chromosome amount, haploid gametes, either eggs or sperms. But there's something special that happens. And when I said earlier that it separates the one from mom and the one from dad, sort of. In this picture, you can see there's 2 colors, purple and green, representing mom and dad, because in the rest of your body right now, the chromosome that you got from mom for chromosome 1 is everywhere, just as you got it from mom. The chromosome 1 from dad, everywhere, just as you got it from dad, with minor exceptions that we won't get into this class. But when it's your turn to have children, to make eggs and sperm, what happens? The chromosome number 1 from mom meets chromosome 1 from dad. They do something called pairing at synapsis. Synapsis is this pairing along their entire length. Kind of a chromosomal intimate moment where they join all the way along the entire chromosome. And in this pairing, they do a process called crossing over. And crossing over, they literally say, I'll give you this part of my chromosome if you give me yours. And they do. They swap chromosome segments. And this is happening for chromosome 1 pairs, chromosome 2 pairs, 3, 4, 5, 6, all the way through to 22. It doesn't happen with the sex chromosomes because they don't line up the same way. But for 22 out of the 23 pairs of chromosomes that we have, synapsis and crossing over. And this re-stirs the pot tremendously and allows huge increases in genetic variation. So you can follow this. I'm not going to go through this in detail. Study this, read up on this, whatever it takes. Talk to the tutor. But this helps walk you through the prophase, metaphase, anaphase, telophase number 1, and then prophase, metaphase, anaphase, telophase number 2, and finally cytokinesis. You end up with 4 cells that instead of having, what did we start with? 4 chromosomes, 2 pairs. Now I end up with 2 chromosomes with only 1 copy of each. And each one of these is a different variation of what I got from mom and dad. They're mixed up. They don't make 4 identical cells. So now with the 4 sperm that came out of this, or the 4 eggs that came out of this single division, what happens? All of them are different because of synapsis and crossing over. And this allows for a tremendous amount of additional variation from generation to generation for organisms that reproduce by sexual reproduction. Huge variation. So there are 2 things that are different between mitosis and meiosis. Meiosis has the synapsis and crossing over. Probably should have changed this slide that I got from somebody else, but should have changed this to synapsis and crossing over. It's part of the same thing. But that's different. Mitosis doesn't have that. You leave the chromosomes you got from mom and the chromosomes you got from dad, you leave them alone until it's time for you to reproduce. Reduction division, where you're cutting the chromosome number in half. That is the 2 things. I ask you on a test, what is the difference, or what are the differences between mitosis and meiosis? Well, mitosis does not have synapsis and crossing over. Meiosis does. Mitosis doesn't have a reduction division. Meiosis does. These are the 2 differences. And here's a picture to help you remember that. What are the differences? The synapsis and crossing over, and the reduction division, where you start with 4 chromosomes. It's actually 2 pairs of chromosomes, but there's still 4 there, even though I've got 2 chromosome 1s and 2 chromosomes 2. As I go through meiosis, I end up with only 1 of each chromosome instead of 1 pair of each chromosome. And here it's lined up, meiosis on the left, mitosis on the right. Here is where we have our synapsis and crossing over. Here's where we have the reduction division. And so they don't have that. During the meiosis 1, or meiosis 1 here, there is no such thing as meiosis 1 in mitosis, because in meiosis 1 is where those 2 differences occur. The pairing, the synapsis and crossing over, and the reduction division, which is occurring right here. So what are the consequences in terms of evolution for sexual reproduction? It's a double-edged sword. Sexual reproduction means you have to have population sizes large enough where you can find a partner and have sex. You've got to exchange genetic material, egg and sperm, in some way. Otherwise, it doesn't work. And so some organisms, it's really tough if you're living in certain habitats. So the clonal reproduction actually has some benefits, but from an evolutionary standpoint, sexual reproduction gives you an explosion of variety that allows your species to adapt to changes in the environment. So one idea is independent assortment. Every single possible combination of egg and sperm cells um, can occur because the chromosomes assort independently. When chromosome 1 from mom versus from dad, they go their separate ways during meiosis. Even though they're mixed up, there's no rhyme or reason where, all right, all the ones from dad move to the right and all the ones from mom move to the left. It doesn't work that way. It's completely independent. The chromosomes assort or move to the poles during anaphase independently. Chromosome 1 separates on its own independently. Chromosome 2 on its own independently. Then there's crossing over and synapsis. The crossing over, that's huge in terms of new variation. And then random fertilization, the idea that any pollen grain has just as much chance as any other pollen grain, or any sperm cell has just as much chance as any other sperm cell. And any egg cell has just as much chance as being the next seed as any other egg cell. And so it's all random. Now we know that this all isn't completely true, but for mathematics and simplicity, let's call it true enough. So let's look at these. But first, another